So let's talk about molecular basis of this genetics, which is coming from DNA structure as a double helix. So this dark and gray ones are a single stranded of DNA, which is supposed to be complementary each other. The important thing is there's a numbering here. That number comes from the numbering of 5 carbon sugar molecule. You see this adenine molecule, adenine with this pentose sugar, 1, 2, 3, 4, 5th carbon. So that from the 5th carbon to the next one connected is 1, 2, 3 prime carbon. So the connection from this one single strand is always 5th prime to 3 prime and keep going until 3 prime. So this we name it as a 5 prime end and this as a 3 prime end. So one is 5 prime end, the other is 3 prime end, and you realize the complementary structure will have to be the opposite way of this. So that's 5 prime end and this is 3 prime end to make this complementary hydrogen bonding. So this structure we call as a anti-parallel orientation. One strand from 5 prime phosphate here to 3 prime hydroxyl. You can see 3 prime hydroxyl. And complementary strand here is oriented in an anti-parallel fashion. So this formed DNA double helix, and the interaction between the different functional groups of DNA bases causes the 2 strands to adopt a double helical structure. And these 2 complementary strands of DNA align such that sugar phosphate backbone. So these sugars are outside, while inside these bases of each strand joined together. And that is the key idea of what's named Greek base pairs. So and let's consider a little bit how this DNA replication can happen. So their physical properties. So native state DNAs are usually double-stranded, and they hold together with hydrogen bond, which is about 1/20th strength of covalent bond. But there are so many hydrogen bonds, so it's like molecular Velcro, and it usually are very stable. But if he added heat or add OH minus, then they can interfere with the hydrogen bond and they can detach, and that we call as a denature, so that we can make this DNA as a single-stranded temporarily. And if you cool down, they actually come back to regip. So that's we call renatured state. And this is very important to manipulate our DNA. So how to separate DNA reversible manner is by detaching this hydrogen bond, which can be denatured by heat. When the temperature is lower, the strands will renature or reassociate according to their ATCG base pairing rules. In fact, this forms a cool idea which led to Nobel Prize to how to amplify DNA. So a molecular biology technique to amplify particular DNA sequence, like exponentially, to study the function of DNA or identify a gene. This is very important technique called the PCR, or polymerase chain reaction. And that also is being used for detecting the coronavirus 19 for confirmation of the patient from their material. So next time we will discuss about in detail DNA replication and PCR further. Thank you for your attention.